Hi, my name is Quinn O'Connor, and I am a senior at Sarasota High School. For my science fair project this year, I am comparing somatic mutations to the Saccharomyces cerevisiae genome when environmental carcinogens are applied. My biggest question was, what effects do environmental carcinogens have on DNA structures and sequences, if any, and if so, what carcinogens have the greatest impact? The purpose of my experiment was to examine the effects of common carcinogens on the genome as cancer rates increase. Since 2015, we have actually seen a 49% increase in cancer diagnoses. For my project, I had 6 different carcinogen trials. I also had a control of 3 different petri dishes. I used UV with protection, UV without protection, thermal radiation with protection and without protection, and then to mimic car exhaust, I used benzopyrene diol epoxide, and to mimic alcoholism and the effects of alcohol, I used acetaldehyde. I did not receive much help from other individuals as I began developing this project in June of 2021 with the original intention of using lymphatic leukemia cells. I searched for a lab from July to the beginning of November, and unfortunately, with a lot of delay, I was unsuccessful in securing a spot. That led me to begin looking for new projects, and I eventually discovered that I could just replace the lymphatic leukemia cells with Saccharomyces cerevisiae. This was a very long and discouraging process trying to find an alternative to my project that I'd worked so hard on, but I'm really happy that I was able to find something that was viable and feasible for my timeline. One of my biggest strengths was how much I cared about this project and how much time I spent developing it, because I did annotate so many articles and do so much research that I really wanted to make sure that all of my procedures were feasible and that they all were actually applicable to real life scenarios. The reason this project was so important to me personally is that I aim to become a pediatric oncologist, and a lot of what I'm interested in is the effects of intergenerational trauma, whether that be physical or environmental. Environmental would be the effects of the somatic mutations due to these carcinogens, whereas physical would be more of the alcoholism and the mutations that those cause. However, I would say one of my weaknesses is my timeline, because I spent so much time fixated on using lymphatic leukemia cells that I was not able to find a lab, and it left me with not a lot of time to get all of my paperwork done and get everything switched over to using Saccharomyces cerevisiae. I actually didn't begin developing my new project, the one that I am currently presenting, until the beginning of December, and I sent out all of my paperwork on December 16th to get approved from SSEA. I had originally not anticipated that I would have to get approval, given that I was using Saccharomyces cerevisiae, which is a relatively harmless microorganism, but because I was opening the petri dishes, I did have to. This was definitely something that was unexpected in my experiment, and that led to one of my weaknesses being that I was not approved until December 23rd, which actually happened to be when we were already on break. I have not yet received most of my materials. I'm very lucky to be working in the MAST lab, where we do have most of what I need, especially because I did do an electrophoresis project last year, but I am still waiting on my Saccharomyces cerevisiae and most of my chemicals to arrive. I ran everything through electrophoresis after each trial. Everything was left with exposure to the carcinogen for 2 hours, and it was then incubated for 2 days. And after those 2 days, I ran it all through electrophoresis and compared it to the control cell and calculated the cell density. Unfortunately, I have not completed my data, but I have put predictions and a timeline in my display. Thank you so much for listening, and I hope you enjoy my project. I'm really eager to get started, and I'm really excited about where I can take this.